Welcome back to the part 2 of Applications of Bioinformatics in Plant Biology lecture. Part 2 will describe about different omic approaches, genome editing, genome sequencing, systems biology, and applications of bioinformatics in plant biology. Bioinformatics today has entered every major discipline in biology. In genomics, bioinformatics has aided in genome sequencing and has shown its success in locating the genes, in phylogenetic comparison, and in the detection of transcription factor binding sites of the genes. Bioinformatics helps in the study of protein structures and the discovery of sequence sites where protein-protein interactions take place. To help understanding biology at the system level, bioinformatics begins to show promise in unraveling genetic networks. Plant life plays important and diverse roles in our society, our economy, and our global environment. Feeding the increasing world population is a challenge for modern plant biotechnology. Crop yields have increased during the last century and will continue to improve by new strategies from bioinformatics and biotechnology. We will see the first application as structural bioinformatics. Protein structure prediction is another important application of bioinformatics. The amino acid sequence of a protein, the so-called primary structure, can be easily determined from the sequence on the gene that codes for it. In the vast majority of cases, this primary structure uniquely determines a structure in its native environment. Knowledge of this structure is vital in understanding the function of the protein. Structural information is usually classified as secondary structure, tertiary structure, and quaternary structure. One of the key ideas in bioinformatics is the notion of homology. In the genomic branch of bioinformatics, homology is used to predict the function of any gene. If the sequence of gene A, whose function is known, is homologous to the sequence of gene B, whose function is unknown, one could infer that B may share A's function. In the structural branch of bioinformatics, homology is used to determine which parts of a protein are important in structure formation and interaction with other proteins. In a technique called homology modeling, this information is used to predict the structure of a protein once the structure of a homologous protein is known. This currently remains the only way to predict protein structures and is more reliable. Now we will see the steps involved in homology modeling. Homology modeling of plant proteins involves the following steps. Selection of protein sequence and family by using BLAST search. Next, the selection of known X-ray structures from protein data bank. Sequence alignment by using CLUSTAL-W. Homology model framework by Quanta. Then the introduction of insertion or deletion of any sequence by Quanta. Add new side chain to the modeled protein by Quanta and Sharam. Introduction of SS ion binding sites by using Quanta and Sharam. Energy minimization by using Sharam. And finally, evaluate the model by ProCheck. By this homology modeling, we can predict the structure of unknown protein. Next, we can see the tools available for plant bioinformatics. The so-called next generation sequencing, that's NGS technologies, as one of the major examples, largely favored deeper insights on plant genome organization and on functional responses to variable environmental parameters, elucidating the first level of gene expression, that is, the transcriptome analysis, by promoting the transition from expressed sequence tags and microarray based techniques to more powerful approaches such as RNA sequencing and associated technologies. Simultaneously, the development of proteomics procedures by 2D gels coupled to mass spectrometry, that's MS, or more recently via high throughput shotgun approaches, and of robust LCMS and GCMS metabolomics technologies are able to unravel fluctuations of non-volatile and volatile metabolites, or paving the way to a better understanding of the effects of the biological processes under investigation. In this context, the integration of results from different levels of molecular information favors holistic views to decipher key components that are playing roles in complex molecular processes involved in plant responses to unfavorable or changing environmental conditions. Moreover, the technological evolution and cheaper methodologies offered faster and more accessible approaches favoring researchers considering an increasing number of crops. Genome sequencing has dramatically evolved in the last years, moving from back by back, that is also known as clone by clone, shotgun strategy, to whole genome shotgun, that is WGS approaches, favored by novel technologies and in particular by the introduction of the next generation sequencing, that is NGS. By using this NGS, we can sequence variety of plant species and genotypes. These efforts provided reference collections, expanding the number of available resources for the same species or for representatives of new species. These efforts are revealing fundamental molecular information that are useful in plant biotechnology and plant breeding programs. Next, we will see on transcriptomics. Transcriptomics is the study of transcriptome, the complete set of RNA transcripts that are produced by the genome under specific circumstances or in a specific cell using high throughput methods such as microarray analysis. Comparison of transcriptome data is highly exploited to understand gene functionality and expression mechanisms in different tissues in various conditions. With the development of the gene chip ATH1 genome array for Arabidopsis, probe sets for around 24,000 genes were designed, disseminating the use of microarray analysis in plant sciences. The Array Express Archive and the Gene Expression Omnibus are main public collections of functional genomics data currently available. DBEST at the NCBI is the specialized division of GenBank which contains all publicly available EST collections from different organisms, currently delivering ESTs from different plant species. The NCBI Sequence Read Archive (SRA) is the reference resource established in order to gather public collections from worldwide. NGS efforts, including data from Illumina Genome Analyzer, Applied Biosystems Solid System, Roche 454 GS System, and Helicos Heliscope. Many transcriptomics efforts also focused on the detection of non-coding RNAs, mainly based on high-throughput sequencing technologies or even on more advanced approaches for their in situ localization. This highlighted the active role played by non-coding RNAs circulating in plant tissues in physiological, pathological, or in stress conditions such as during other stress conditions. Small non-coding RNAs of 19 to 23 nucleotide length under abiotic stresses in many plant species are analyzed. miRNAs are involved in stress signaling pathways responsible for root development, leaf morphogenesis, and stress responses, and also it crosstalk and interact with phytohormones signaling pathways. Small RNA collections are also publicly available in RFAM Mirbase databases. Examples of other dedicated platforms for related analysis are Plant Dario and PS Robo. Targeted modification of plant genome is a key for elucidating and manipulating gene functions in basic and applied plant research. The CRISPR-Cas9 technique is widely adopted for genome editing in plant biology. The CRISPR that is clustered, regularly interspaced, short palindromic repeats and CRISPR associated protein, that is Cas technology, is emerging as a powerful genome editing tool in diverse organisms. This technology utilizes an easily programmable guide RNA, that is gRNA, to guide Streptococcus pyogenes Cas9 endonuclease to generate a DNA double-strand break, that is DSB, within an intended genomic sequence and subsequently stimulate chromosomal mutagenesis or homologous recombination near the DSP site through cellular DNA repair machineries. Several software have been developed to predict and select CRISPR-Cas9 targets for genome editing in plants, such as the web-based tools eCRISPTide, ChopChop, and CCTop. Next, we will see on phylogenetics and its applications in plant bioinformatics. Phylogenetics is an attempt to reconstruct the evolutionary relationship between different species of plant and also their origin. Historically, this was done using quantitative morphological data based on the external appearance of the plants. However, the modern methods include the DNA sequences and it rely more on DNA sequences data to construct the phylogenetic tree. Here you can see the picture of phylogenetic tree that is constructed for various plant species. Here we can see different phylogenetic tools that are available for plant bioinformatics. The first one is MEGA, that is Molecular Evolutionary Genetics Analysis. It's a computer software for conducting statistical analysis of molecular evolution and for constructing phylogenetic trees. It includes many sophisticated methods and tools for phylogenomics and phytomedicine. The next one is Phylogeny Inference Package, that is PhylIP. It's a free computational phylogenetics package of programs for inferring evolutionary trees, that is phylogenies. It consists of 35 portable programs. Next one is QuickTree. that is also used for construction of phylogenetic tree and optimized for efficiency. Then the next one is partition finder. Combine the selection of models of molecular evolution and partitioning schemes for DNA and protein alignments. APE is based on R program and it's a R project package for analysis of phylogenetics and evolution. Next one is systems biology. Systems biology is an approach by which a system of interacting units is analyzed as a whole rather than by analyzing its individual constituents separately. Currently, there are three types of systems approaches exist, such as top-down, which entails the use of high-throughput omics technologies to gain a holistic understanding to biological systems. Bottom-up, which starts with molecular properties and leads to derivation of models to be tested and validated. Middle out, which is based on the principle to start somewhere in between the top and bottom levels, then work out toward a hierarchy of models. Amongst these, the top down and bottom up have been most usually used to describe the biological systems. Various tools are available to work on systems biology. For example, for visualization of omics data on a systems scale, SunGear, MapMan, GeneVestigator, Cytoscape, Virtual Plant and Reactome are available. If anybody wants to study on pathway, then pathway database for system analysis includes KIG, BioSeq, ARASeq, pathway interaction database, that's PID. Then comprehensive modeling environments include Virtual Cell or MATLAB Systems Biology Workbench. Then model repositories include Biomodels Database or JWS. Then core system biology networks include SynBio, WaveCell, Memosis, Bablomics and MetNet. Hence, systems biology will give rise to immense opportunities in decoding the complexity of plants by the fruitful collaborations of the classical plant biologists and computational modelists. The unity in diversity approach of systems biology is growing and will continue to impact the remarkable future of plant science, thereby getting the most of the plants worth to benefit humanity in a short time frame. Tools of bioinformatics are playing significant role in providing the information about the genes present in the genome of these species. These tools have also made it possible to predict the function of different genes and factors affecting these genes. The information provided about the genes by the tools makes the scientists to produce enhanced species of plants which have drought, herbicide and pesticide resistance in them. Certain changes can be made in their genome to make them disease resistant. When the evolutionary changes occurred in the plants, their genome remained conserved and did not provide much information. Since the arrival of bioinformatics tools, it's possible to extract the required information from the genome of specific plants. There are few plant species, the genome of which has been mapped completely, for example, Arabidopsis and rice. Arabidopsis is a small plant which is found on the rocks. Researchers took interest in its genome because it's a smaller genomic size and studied the plant developmental processes. Its genome consists of 5 chromosomes and is 100 megabase base size. It reproduces within 5 weeks and makes next generation. The understanding about its genes and their expressions provides information about other plants, proteins and their expressions. There are many uses of knowing the genome of Arabidopsis, but The major use is that the yield of the plants can be increased by using this Arabidopsis genome. Many plants have been made insect resistant by incorporating the desired genes. Bacillus thuringiensis is a bacterial species which increases the soil fertility and protects the plants against pests. When the researchers mapped its genome, they used its genes to incorporate into the plant to make it resistant against insects. For example, corn, cotton, and potatoes have been made insect resistant so far by using Bt. By having the genes of bacteria in the plant's genome, when insects eat the plants, the bacteria enter in their bloodstream and make them starved. Ultimately, they die. Bt corn is one species of food plants which have been modified by inserting bacterial genes in it. It's effective against insects by developing resistance against them. The use of Bt genes in the plant's genome has made the agriculturists to use the insecticides in very little amount. As a result of this, the productivity and nutritional value of plants also increased. That will benefit the human health. When the changes are made in the genome of the plants, the nutritional value of plants also increases. For example, some genes are inserted in the rice genome to increase the vitamin A level in the crop. Vitamin A is an important component for the eyes, and if the vitamin A deficiency occurs in the body, it may result in blindness. This work has allowed the scientists to reduce the rate of blindness from the world by giving genetically modified rice to the people. Some varieties of rice are also developed which have the ability to grow in poor soils and are drought resistant. From this lecture, we can conclude that bioinformatics can be applied in plant biology to identify novel plant species based on the sequences, and also we can identify the evolutionary relationship among the species by constructing phylogenetic tree, and also we can identify the functions of proteins and enzymes and also the unknown protein structure by homology modeling. By using CRISPR-Cas9 genome editing technology, we can produce crops that are resistant to pathogens and resistant to various diseases and environmental conditions. Metabolomics approach will be helpful to produce novel metabolites by engineering the plants by using bioinformatic tools. NGS will help to sequence more plant genome for the benefit of human beings. Altogether, bioinformatics will be very helpful for sustainable agriculture. Thank you very much for your time.